Hello, friends of the sun, and welcome to today's reading group where we're going to discuss MFBind, a new multi-fidelity approach that combines experimental data, docking data, and free energy simulations to build a more powerful and more generalizable model.
Thanks for the invitation. I'm excited to talk today. So yeah, I'm Peter Eckmann. I'm an undergraduate at UC San Diego studying computer science. And today I'll be talking about our recent work, MFBind, which is a multi-fidelity approach for evaluating drug compounds in practical generative modeling. And this work was done in the lab of Dr. Rose Yu, who's a faculty of machine learning at UC San Diego. And we were also fortunate to be able to work with Dr. Michael Gilson, who's a faculty at the School of Pharmacy, also at UC San Diego. So for some background, generative models for de novo drug design kind of have been showing promise for accelerating drug discovery for the past few years, but they're not yet really commonly utilized in real-world drug discovery projects. And the central reason for this is that the computational prediction of binding affinity remains difficult. So molecular docking is the prevalent technique used by these generative models due to its speed. So molecular generative models that utilize reward functions, such as like reinforcement learning-based techniques, VAEs, genetic algorithms, all use docking because it's required to have fast evaluation of compounds. Structure-based algorithms that generate a 3D molecule given like a protein pocket, such as like some of the diffusion models, do not directly require a reward function, but they still often use docking in kind of like a post-processing step to rank the generated compounds and kind of get the compounds that are most promising. But the big problem with this is that docking is generally kind of inaccurate. So even generated compounds that have high docking scores are unlikely to show binding, which means that the compounds generated from these models are also unlikely to show binding. But there are more accurate techniques to computationally estimate the affinity. So in particular, binding free energy techniques utilize a series of molecular dynamic simulations. And these techniques are estimating affinity a lot more accurately than docking. So on the right here, we just kind of have an example of how to calculate the binding free energy. So each circle here kind of represents a molecular dynamic simulation that you have to run. And we first kind of start with just the ligands in interacting with water, and it's important to model all of the water molecules kind of explicitly. And then in a series of steps, we kind of first turn off the interactions of the ligand with the environment. We attach the protein to the ligand, and then we kind of turn back on the interactions to get this final complex of the protein and ligand in interaction with water. And then, through these calculations, we can calculate the delta G, which is the binding free energy. So this process is a lot more, involved than docking, but it's also a lot more accurate. But the big problem with this is that the calculation of a single protein ligand pair can take hours to days to simulate because we have to run so many molecular dynamic simulations. And for that reason, it's not really feasible for use in a generative model because we have to evaluate, thousands or more compounds in a generative model. And it's just not feasible to run that much using this technique. And kind of in a separate thread of the background, we'll also talk about multi-fidelity learning. So multi-fidelity learning aims to fuse multiple data sources of variable accuracy and cost to achieve an optimal trade-off between accuracy and cost. And it involves usually modeling each fidelity with the help of some shared representation across all the fidelities. So on the right here, we kind of just have an example of what a multi-fidelity environment looks like. So the x-axis is just some input variable, and the y-axis is some response variable. And we can see the ground truth is kind of the dashed line here. So that's what we're trying to simulate, or that's what we're trying to predict. And we have some high fidelity simulator, which are like these orange dots here. So you can see they closely match the ground truth. And this would presumably be the expensive simulator. But then we have some cheaper simulator, which is the low fidelity in blue here. And we can see maybe there's like more noise added and it might be shifted from the ground truth, but it's still, potentially helpful in the prediction of the ground truth. So, we can have a model that queries a mix of these two to kind of make the most accurate prediction using the fewest number of calls. So for multi-fidelity learning, GPs are often used, Gaussian processes, because of their data efficiency and their well-calibrated uncertainty estimates. And multi-fidelity learning is commonly applied to problems in climate modeling and fluid dynamics, but the previous applications in drug discovery have been somewhat limited. Like there's been some examples of, you train multiple models maybe on a subset of data or something to kind of simulate something that's low fidelity. But so far, it hasn't, like no one has really utilized some real simulators and real costs and applied it to this multi-fidelity modeling. So our study goal was to combine docking and binding free energy techniques using multi-fidelity learning to achieve the optimal trade-off between speed and accuracy. So this is just kind of an overview of the multi-fidelity bind framework, or MFBind. And this is what I'll be talking about mostly. And I'll break down this figure individually in the next few slides. But just in general, here are our simulators. So we have molecular docking and binding free energy. And they take a compound as input, and then it gets run through either of these. And then the output value gets kind of added to a data set in combination with, experimental data as well. And then all this data is used to train a multi-fidelity model. And then this multi-fidelity model is used to evaluate something called an acquisition function, which tells us what compound to query the simulators with next. And then simulators get run again, and it kind of repeats in this cycle. So first, we'll talk about the environment, which just consists of these simulators and the experimental data. So we defined a multi-fidelity learning environment consisting of 3 fidelities. So our lowest fidelity is Autodock 4, which is a docking program. And that takes about 30 seconds per compound to run. Our middle fidelity is experimental data from binding TP, which is our middle fidelity. And then our high fidelity is absolute binding free energy calculations, or ABFE, which is a type of binding free energy calculation. And that takes about 10 hours per compound. And the reason we have ABFE as the highest fidelity, although you might expect experimental data should be the highest, we want our highest fidelity to be kind of queried. We have to be able to query it for any arbitrary compound. But we can't do that with experimental data because it's just like a fixed set that we have available. So although technically we want to, generate compounds with good experimental binding, for the purposes of this environment, we kind of treat ABFE as the highest fidelity and experimental data as the middle fidelity. Although, in the real world, I guess we'd want to reverse the two. So our experiments on the right confirmed that ABFE is significantly more accurate at separating the true actives than docking. So you can see we have a ROC curve here for BRD4, which is one of our test targets. And we have ABFE shows, strong binary classification performance, but docking, and you can just look at the orange line for now. Docking shows, not as good classification performance, but it still has some predictive value. So for this reason, this is kind of a good setup for multi-fidelity learning, because we have some high fidelity simulator that's accurate but expensive, and then we have some lower fidelity simulator that's not as accurate, but it still has some predictive value. So now we'll talk about the multi-fidelity model in our framework. So we designed a neural network-based surrogate model to learn a cheap approximation of the highest fidelity, which is ABFE, using data from all the fidelity levels in our environment. So we have a deep neural encoder to generate a representation of the input molecule, and then fidelity-specific linear heads to produce an output at each fidelity level. So you can see on the right here, we kind of have a diagram of the surrogate model. So it takes a Morgan fingerprint as input, and then it gets fed through some deep neural encoder here to generate some latent representation. And then this representation is then fed to each of these linear layers here that produces a prediction of the output at each fidelity level. So we have, one layer to produce one for autodoc, and then we have one for each other fidelity.
Yeah. We've got a question from the chat, which is, I guess, does the ABFE simulation require some initial docking pose from, say, your docking algorithm?
Mm-hmm. Yes, that's a good question. It does require an initial pose from docking. So we use the pose generated by autodoc to kind of seed the ABFE calculation. But even with that, ABFE is a lot more accurate than just docking. So you could consider, I guess, ABFE some kind of docking rescoring method. But yeah, that's a good question.
Mm-hmm.
So I guess back to the okay. Sorry. Back to the surrogate model here. So training the surrogate model on all the fidelity levels kind of allows it to learn this shared representation. So it'd be this encoder here. That's useful for the prediction at all the fidelity levels. And just some key differences between our and some previous multi-fidelity surrogate models include, first of all, regularization of the fidelity-specific prediction heads to avoid overfitting on the small amount of high fidelity data that we have. So we have, less than 100 samples of high fidelity ABFE data to train on. And we found that without this regularization, we have the issue of, overfitting this kind of prediction head here, which led to poor generalization. But if we kind of enforce some regularization on this linear head, then the generalization gets a lot better. And the second technique is a pre-training technique. So this involves first kind of training on a lower set of fidelities, and then subsequently on all fidelities. And this is also to address the overfitting issue. So this involves kind of first training the model on these two lower sets, or this set of two lower fidelities, autodoc and experimental data, and then subsequently on all three. And the kind of reasoning behind this is that without pre-training, we found that trying to predict the small amount of ABFE data that we have, so less than 100 samples, kind of overly influenced the compound encoding, which led to poor generalization. So I guess you can think of it, we have overfitting not just at the kind of fidelity-specific prediction heads, but also we overfit kind of the encoder because we have so little ABFE data. But when we added this pre-training method, we pre-train the model on this large amount of lower fidelity data that we have. And that kind of gives us like a warm start to the encoder because it generates a more meaningful encoding such that when we add back in the ABFE data at our later stage of training, the generalization was a lot better. And the third difference, the third main difference is the ability to output very dimensional outputs at each fidelity to take advantage of the multiple quantities generated by autodoc. So autodoc doesn't just output kind of an estimate of the binding energy, it also outputs a bunch of other values associated with binding, such as other components of the binding energy or number of hydrogen bonds, that sort of thing. And we found that if we model all of these simultaneously, using our multi-fidelity surrogate, the performance actually improves. So finally, in this architecture, I'll talk about the active learning framework. So to train the multi-fidelity surrogate model most efficiently, we proposed an active learning algorithm to iteratively query the simulators on the molecules that the model is most uncertain about. So the idea here is that we have, a set of kind of candidate molecules, and then the active learning algorithm selects which molecules to query the simulators with, and then we run the simulators on those molecules, and then add that to the training data set, and then repeat the process. So for this, we need an acquisition function, which is a of x and k, that returns kind of the expected utility of querying a simulator for a molecule x at fidelity level k. So this equation here is just how we define the acquisition function. And sigma squared is the posterior variance of the model, and c sub k is the cost of querying simulator k. So I guess this first term here, which is 1 over c sub k, makes it kind of cost weights the acquisition function. So if we have a more expensive simulator that takes more time to run, the acquisition value will be lower, which is what we want, because we don't want the function, constantly querying the high accuracy simulator. And then the sigma squared, which is the posterior variance, essentially, we use Monte Carlo dropout to estimate that. So that means we give the model kind of the same input multiple times, and we train it with dropout. Is that a question or? Okay. So we use Monte Carlo dropout to estimate the posterior variance, meaning we give the model the same input multiple times, and then just compute the variance of the output, with the model having dropout. And the idea here is that, the higher the variance, the more uncertain the model is. So these two terms kind of, I guess, clash with each other. And whatever molecule and fidelity pair that has the highest value is the one that gets chosen to query the simulator. And this is just, the summary of the algorithm. So the active learning algorithm requires some surrogate model G, training data set D that's pre-populated with some set of values, a set of candidate points S, which means that the set of compounds that are kind of available for the active learning to choose from, and run the simulators on. And then we have some, cumulative cost of active learning C, and then some budget B. So in general, the algorithm, so while the, running budget is less, or while the running cost is less than the total budget, we can first train the surrogate model, or we first train the surrogate model on the data set, and then we iterate through the candidate points and each fidelity level, and we evaluate the acquisition function at each value. And then we take the highest value of the acquisition function, and then take that compound and run the simulator on it and append those results to the data set. So that's this line, where we just append the results of the simulator to the rest of the data set. And then we also increment the active learning cost with the cost of the simulator. So in summary, the MFBind framework consists of a multi-fidelity environment, surrogate model, and active learning algorithm. So I guess I'll just kind of go through this figure again now. So we start with some acquisition function that, we evaluate on all our candidate compounds. We take whatever compound and fidelity level pair that has the highest acquisition value, and we query the simulators with it. So we can see that, docking has more speed, but is less accurate. So we let the model kind of choose what to query here. And then we take that data and append it to the data set that we have, which includes experimental data, and then use it to train the multi-fidelity model. And then after we train the model, then we can use it to reevaluate the acquisition function, and the process repeats. So I guess now we'll talk about the results. So we first wanted to evaluate the surrogate modeling performance of MFBind. So to do this, the first step was to train MFBind and the baselines using the active learning algorithm that we just talked about. So to do this, we first supplied each method with an initial data set of 100,000 docking values, about 100 experimental values from binding GP, and also one ABFE value. So that was kind of our initial data set. And then we ran active learning for 8 days, starting from this initial data set. Is it possible to mute the typing? I'm not sure who that's coming from.
I think, yeah, I think that's Mike Gilson.
Oh, okay. I think it.
Yeah, my bad. Sorry about that.
Okay, no problem.
Yeah, thank you.
So the first step is to train MFBind and the baselines using this active learning algorithm. And then after we ran active learning, we measured the performance in mean squared error on a held out test set of pre-computed ABFE values. So this test set consisted of 32 compounds, half of which are known actives for the targets we looked at, and the other half are presumed decoys. So that just means, we take some compounds that are similar in properties to the actives, and we just assume they're, inactive because they're randomly selected from some big data set. And we kind of made the test set like this so that we had a good range of activities. So we both had, highly active and inactive compounds to test the model with. So we performed evaluations on 2 separate targets. So the first target was BRD4, and the second target was CMET. So we picked these targets because they're well studied with ABFE, meaning we know that the ABFE is well correlated to experimental values with these targets. And they also have large data sets available. So meaning, we have lots of experimental activity data for these 2 targets. And, the targets are also kind of substantially different at the molecular level, which gives us a good, testing case for our model because we have 2 pretty different targets. So BRD4 is bromodomain containing protein 4, and it binds to chromosomes during cell mitosis and helps to regulate cell growth. And its inhibition may be helpful for reducing cancerous cell growth. And CMET is a receptor tyrosine kinase, which is involved in many cellular pathways, including cellular growth. And it may appear in many types of cancers and generally carries a poor prognosis. So both of these targets are relevant, mostly to cancer, but they're both, well studied in drug discovery. So now for the actual results, we compared the performance of MFBind to a simple single fidelity approach that just involves training a neural network surrogate on only ABFE data. So that's the orange line here, which is like only ABFE neural network. And that kind of represents the, I guess, paradigm of just using some single fidelity simulator. And then we also compared to multi-fidelity, to other multi-fidelity active learning techniques. And these are both GP-based, which is, again, Gaussian process. So that's the surrogate GP, direct GP, and Hadamard MT baselines. And we also compared to some neural network-based multi-fidelity active learning techniques. So this is DMFDAL and DMFAL. So these are the results here. So on the x-axis, we have the cumulative cost of active learning. So as we go to the right, we spend more time on active learning, which means we've queried the simulators more. And the y-axis is the MSC on the held out test set. So a lower value here is better. And we find that MFBind achieves the lowest MSC, so measured in kcal per mole, on this held out test set of ABFE computed compounds. So we can see that MFBind kind of generally most efficiently uses the active learning budget to make good predictions on the test set.
Yeah, I had a question about these graphs. I guess what
Yeah.
was the variance of the data or the kind of, I guess the holdout set? Those MSEs are pretty large. That I guess for all the models, I wonder.
Yeah.
Yeah.
So the shaded region here is the it's the standard deviation over 20 runs with random seeds. So I guess that kind of directly answers your question, but the MSE is, I guess, kind of seems generally high, but I would say at least for BRD4, it's kind of in the useful range already. So, you know, an MSE of 20, given that, you know, the difference between active and inactive compounds can be, you know, 5 to 10 kcal per mole. I think this MSE is still, you know,
Yeah.
about on the verge of being useful, I would say.
Yeah. I guess I was thinking. I wonder, like if you predicted.
Yeah.
I wonder if like, did you look at, I guess, the correlation coefficient there?
Yeah, I looked at that as well. That's about, you know, 0.3 or 0.4.
Okay.
But we don't have graphs showing that, but yeah.
Okay. No, I think, yeah, I guess I was kind of thinking with some of the high MSEs, it might be a sign that all of them are kind of offset by some amount, but which is fine for kind of virtual screening when you mostly care about correlation.
Yeah, I mean, that's a good point. That's possible. For both of these targets, it was about, you know, 0.3 or 0.4. But yeah, I think the CMET MSE looks higher because the range of possible activities is a lot higher. I think, is there a question from Dominique?
Yeah. So when you say 0.3 or 0.4 correlation, like which correlation metric are you using?
Just the Pearson's correlation.
Okay. Okay, cool. So yeah, it's not such a great correlation, but it's not. It doesn't like it still shows a bit of signal, I guess. I'm wondering, like, in the methods that you compare here, like these are like for free energy prediction, but have you compared, like, for example, ranking of compounds between your model and like docking?
We didn't directly compare with docking in this experiment, but in a subsequent experiment we have those results. So I'll talk about that in a bit.
Okay, yeah, eager to see those results. Yeah, thank you.
Yeah.
Hassan, you have a question as well?
Yeah, I guess it's the same vein. You know, like any ranking correlations for these data sets, like Tau or Spearman? Because I feel like for
yeah.
oh, sorry.
Oh, I mean, yeah, I didn't look at that, but
yeah,
I think that's a good idea. Yeah.
Yeah, because I feel like for absolute binding free energy, I mean, what you're really interested in is saying whether one compound binds better than another, or like if you're doing like lead like compound optimization. So yeah, I mean, and then if you're also testing, so all of these are on the same receptor, right? Wouldn't it be faster to do relative binding free energy calculation, or look at that?
Yeah. So I guess the reason we don't do relative binding free energy is that we're testing this on for like de novo discovery. So relative binding free energy requires that you have some known active, and you know it's like experimental affinity, and then you can look at the, you know, you can predict the energies of compounds similar to it, but we don't assume that we have that data. And I guess another reason is that we don't have necessarily congeneric ligands here. So I'm not sure if relative binding free energy can really apply.
Oh yeah, that's fair. That makes or well,
yeah,
I guess
that's a good
I don't
question.
know how hard it would be to do pairwise FEP for non-congeneric ligands. But, okay, thank you.
Yeah. So I guess I'll keep going.
Yeah. I have another question. Like,
yeah.
maybe you said it, but like when we look at binding, we care about like the difference in free energy between like the unperturbed like protein and the one with the ligand. So uh, did you look at the difference, or like just here at the MSC between the actual free energy and the um
You mean so like when we compute the binding free energy, you mean? Like are we
I mean
computing the
yeah, it's because like free energy like
Mhm.
the protein itself has a certain free energy.
Yeah.
And like I wonder here if you looked at the difference between the initial free energy of the protein and the one where the ligand is introduced, or like you're looking just at the uh, absolute like free energy of the system.
Oh, yeah. So we look at the difference in the free energy between the bound complex, which is
Yeah.
the protein, the ligand. And I guess, you know, the difference between that and the energy of just the protein by itself.
Okay,
So
cool.
so
Yeah,
yeah,
you
we're looking at the
probably
difference.
okay. You probably mentioned that earlier, so sorry. I
Yeah,
asked
no
you.
problem. Yeah, so I guess I like the idea of potentially looking at the rank correlations as well. I think that's yeah, we have a few questions about that, but I think that makes sense. So that's something I'll look at. So so we have the surrogate modeling results on the baselines. And I also looked at so we also looked at the impact of various ablations on our approach in terms of the test set MSE. So on the right here is we just kind of have a table of the different um, ablations that we perform on our architecture. So we showed that both the pre-training technique that we have and the regularization of the prediction heads is important for the model performance. So you can see for both of these targets, the MSE is higher for, if we remove these two techniques. And we also found that removing either of the lower two fidelity levels, so Autodoc4 or the experimental data, we found that hurt performance. So you can see, without the Autodoc data and without the experimental data, the MSE is higher. So that kind of shows that all fidelity levels that we have are important for the prediction. So next, after the kind of experiments on the surrogate modeling, we aim to evaluate MFBind in a generative modeling experiment. And we got a question about what regularizations were used. The regularization we did on the prediction heads is just the L2 regularization. And we also got a question about if we assigned a time cost to querying the experimental data. So I should have mentioned this, I guess. But the experimental data was not a simulator we could query, I guess. So we could query the docking and ABFE simulators, but we kind of treated experimental data as this fixed set of compounds, and we can't query more. So that was just kind of added to the training data set to help in prediction. But yeah, it's not something we can query for arbitrary compounds. So we only allow the model to query, from docking and ABFE.
Yeah, I guess does that mean you kind of used it like with the like pre-training set of the Autodoc
Yeah.
data? Okay.
Yeah. Okay, so yeah. So next we aim to evaluate MFBind in a generative modeling experiment. And this is kind of where we compare it with docking. So we tested MFBind in a generative modeling setting by combining it with our previously developed generative model, which is Limo. And that's a VAE-based generative model. And the way we combined it is just that we use the predictions from MFBind as kind of a reward function for Limo. So this is kind of an overview of the Limo architecture. I won't go into too much detail, but we have regular kind of VAE setup. And then we have some property predictor. And then we backpropagate the results, or we backpropagate the output of the property predictor back to the latent space to kind of optimize the molecules. And essentially just what we did here is we used the MFBind predictions as kind of another target property. So the limo model optimized the MFBind predicted affinity. So we compared the MFBind model trained using active learning. So we used the active learning from the previous section to single fidelity or SF methods that involve training a surrogate on either only ABFE or only AutoDoc4 data. So I guess this is the kind of most direct comparison we have to docking. So to do this comparison, we generated 20 compounds from each method and then used ABFE to estimate their affinities. So this table at the bottom here shows the results from this. So we report the mean as well as the top 3 compounds out of the 20. And these are all kcal per mole values from ABFE. And we do this for both BRD4 as well as CMET. So you can see from MFBind both the mean energy of the compounds that we generate as well as the energies of the top compounds are a lot more favorable than the single fidelity methods. So, I guess single fidelity AutoDoc4 is just, you train a surrogate on only docking data and then use that to generate compounds, and same for ABFE. So yeah, MFBind plus Limo generated compounds with markedly higher ABFE predicted affinities than the single fidelity approaches, which shows the advantage of the multi-fidelity approach in this generative modeling setting. And I guess to get a sense for what kcal per mole values are good, generally less than -8.2 kcal per mole means the compound's in the nanomolar range of activity, which is kind of a generally desirable range for early stage drug discovery. So I think the activities of these compounds is generally about in the range that one looks for in a early stage drug discovery project. And single fidelity approaches don't get too close to that. And here are just some examples of the generated compounds from MFBind and Limo. So this is these are the same numbers as the table, but just these are the actual compounds. And you can see the ABFE computed affinities down here. So, in general, I think they look generally kind of drug-like. And we didn't, directly evaluate the synthesizability, but I think, these compounds look sort of reasonable, and generally look drug-like. So yeah, so for conclusions, MFBind outperforms the single and multi-fidelity approaches in surrogate modeling and compound generation challenges. And crucially, it looks like MFBind combined with Limo generates compounds that have much higher ABFE computed affinities as compared to the single fidelity approaches. Some limitations of this approach include using a limited set of fidelities. So we only have 3 fidelities here, but you can imagine adding more. And we didn't really directly account for the synthesizability of the generated compounds. So I think if you wanted to apply this to a kind of more realistic drug project, you'd want to somehow take into account generated compounds that are synthesizable. And future work includes expanding the set of fidelities that we have in our environment. So you can imagine using maybe ML-based binding affinity predictors as well, or maybe other docking programs. And then, hopefully, eventually experimentally testing the generated compounds. So yeah, thank you all for your attention. We have the work is available on arXiv right now, and it's currently undergoing peer review. So I guess if we have any more questions, I'm happy to answer them.
Thank you, Peter. Definitely very impressive results at the end. Dominic, you have a question?
I was clapping. Was
Oh, okay. Sorry.
But I have a few questions, but let's you first if you have any. No? Okay, so let me go first then. Well, thanks a lot for the presentation. It was super interesting. Just like here you mentioned the multi-fidelity where you mostly use like docking and binding free energy like to train the model. And I'm wondering like one of the things that's interesting with multi-fidelity is like to really evaluate how much data of each fidelity is needed to like achieve like the final performance. And I'm wondering like if you had some kind of ablation like that where you would take only like half of the low fidelity data or half of the high fidelity data, or even less than half, like 25% of that, and seeing like, okay, well, to make the model better, what is missing? Is it the low fidelity that's missing, or is it the high fidelity that's missing?
Yeah, that's a good point. We didn't directly look at that. I guess in terms of your question about how much data was queried, I think it was about proportional to the cost, I guess. So we have, maybe 100 times more docking data queried than ABFE or so. But yeah, I think looking at, if we only take a subset of some fidelities, I think that'd be a good experiment to do. So thank you. I think we'll look at that.
Yeah, thanks. And your models were trained like you only have a few proteins, I think, on which you show the results, right? Yes. So was the model trained multi-target or was like many single target models?
It was single target. So we trained a separate model for each different target.
Yeah.
Yeah, I mean, expanding to multi-target, I think would be a good future work. And we'd also like to look at it, I guess, look at the results for more than just 2 targets. But for now, we were kind of constrained with what we have ABFE kind of set up for.
Yeah, and was your model just a model like trained on the ligand, or was it trained like somehow with the some sort of protein embedding as well?
It was just the ligands.
Okay, so yeah, I guess that's why. Well, if you were to have a multi-target approach, then you'd have to embed the target in some ways. Okay,
Yeah.
cool, but very, very, very interesting work.
Thank you.
Thanks a lot.
Thanks.
Thanks. Azam?
Oh, yeah, I guess. I don't know if you talked about this, but I was looking in your paper about the ABFE calculation. So you use this automated pipeline like bat.py, right?
Yeah.
And do you use double decoupling or do you pull it from?
We use the SDR method, which is the simultaneous decoupling and recoupling.
Okay. So I guess for the simultaneous one, from what I'm familiar with, is usually turning off electrostatics and then van der Waals. How many did you have any like issues like when you're automating this, kind of like running into ligands that explode or anything during the course of the simulations, or how robust are these calculations?
Well, I guess I think there's some papers already about ABFE for that particular target. So I guess we can say the calculations are pretty robust.
I meant like while you're doing the active learning, is there like a failure rate for compounds?
Oh, I see.
Like how much time is wasted in these? Like if you start a simulation, you're like, oh, I can't finish the calculation or something.
Yeah. So generally, for active learning, we always kind of wanted I guess some binding energy output. So you can sometimes terminate the experiments early if you find that like the ligand is kind of drifting away from the binding site. But we didn't do that for this experiment. We just kind of ran it no matter what to generate some binding value.
Yeah, that makes sense, because I guess something that you could in the future add is check the convergence, like while you're doing, or check the overlap between like lambda values in your, I don't know if they're using TI or FEP or whatever, but
yeah, I think TI, but yeah,
okay.
That's definitely a good idea. Yeah,
Yeah.
thanks.
Oh yeah, Oliver.
Hey Peter, hey Mike, very neat work. I think
Hi.
I'm kind of just hey, I'm kind of like interested in how you utilize those chemical data that you generated, because I remember like early on you showed a figure that you had like AutoDock4 with only binding energy and AutoDock4 with all outputs. I was kind of wondering like what are the additional information that you were using there?
Yeah, that's a good question. So we experimented with both just using the kind of total binding energy prediction from Autodoc, which worked fine, but we found it worked even better when we add, a bunch of other values, because it's just, more things that the model can use to help predict ABFE. And the full list, I guess, of different outputs that we use is in the paper, but generally we used kind of the different energy components of Autodoc. So Autodoc outputs maybe 4 or 5 different values it uses to compute the total energy. Like I think this, like a van der Waals energy, and there's a bunch of different ones. So we used all of those, and then we also used some other information about the interactions, like the number of hydrogen bonds, the number of like T-stacking interactions, the number of close contacts, all of those things. And then we also looked at, there were some values about the sidechain flexibility from docking that we used as well. So yeah, it's kind of just a mix of a bunch of different random outputs from Autodoc, but we found that, using all of them or trying to predict all of them led to increased performance. So we kept them.
Yeah, that's very interesting. And for the ABFE calculated data, did you just use sort of like the final energy output as like get fed into your machine learning model, or did you also use some trajectory related data?
Yeah, so for this paper, we just use the final energy output, but it's definitely, I guess, a potential future work to take advantage of all the outputs from ABFE as well, and model all of those. So yeah, I think that's a good future direction.
Awesome. Yeah, I think as you'll have some like other maybe question or discussion, I'll probably just like email you later on to have the
Yeah, that's
discussion.
fine.
But super nice work. Yep.
Yeah, thank you.
Mayank, is that how you pronounce your name?
Yeah, Mayank.
Mayank. Okay.
Yeah. So, going on the thread of Autodoc, so from a computational perspective, did you use a GPU version or you?
Yeah, we used Autodoc GPU, which is a GPU accelerated version of Autodoc 4.
Okay. Okay. So, did you guys had some benchmarks, or did you had some compute related how do I put it what hardware did you actually use? Did you use A100s or?
I think it was 2080s. I'm not
Okay.
sure exactly.
Okay.
I think
Okay.
it's in the paper, but we have a server of 8 GPUs.
Okay.
I think it was 2080s, but I'm not sure. I need to check again.
Uh-huh.
But
Okay.
yeah, I
Thanks.
mean that's definitely that's definitely relevant. Because if you use, a more modern GPU, the speed at which you can run ABFE and docking is like can be improved by a factor of 2 or so. So yeah, that's a important question.
Yeah. Thanks.
One question I had is kind of about the how important was the experimental data? Did you do, I guess, ablation tests with and without it?
Yeah, so
Yeah.
that'd be this one here. So this
That's
last
the
row is when we train MFBind, the same as the regular MFBind, except we just get rid of the experimental data.
Cool. Yeah,
So
definitely. Yeah, it looks like it's, I guess, maybe more valuable than the AutoDoc data.
yeah,
I mean, it's
that's
hard to
a
make.
yeah. I mean, it looks like that. Yeah, I think comparing the relative importance of the two different data sources makes some sense to do. But yeah, I mean, it seems that both help to some extent, but yeah, experience data seems to be more important than AutoDoc.
Yeah.
But Peter, so I ought to know this, but the ultimate MSC is not against experiment, right, that you're reporting, right? It's against more ABFE, right? Data.
Yes.
Yeah, and on the note, some of the pretty impressive binders you're generating in your generative step, I guess in your generative experiments. I wonder how far away they are from the experimental data that was fed in them. Are they pretty novel, or are they somewhat similar?
Yeah. That's a good question. So we looked at the Tanimoto similarity between the generated compounds and the experimental data, and they all, they're like relatively dissimilar. So the Tanimotos are less than 0.3 or so. But yeah, that's absolutely something that's important.
Yeah,
You
cool.
there's
But
another
yeah.
paper. I think Peter's first paper on limo probably really goes into that, right?
Yeah, well, I guess the relevance here would be that since we train MFBind on the experimental data, if it just predicts the compounds it's already seen as highly active, then that wouldn't be too helpful. But yeah, it seems like there's some significant dissimilarity between the generated compounds and the experimental data.
Yeah, it's really cool. I mean, it's like the tightness of the binders you're generating, it wouldn't seem like you would find them kind of by random chance when you're querying the ABFE model. So it's definitely, it makes it look like the model is generalizing, which is cool.
Yeah, I agree.
Yeah, I guess any final questions before we tap out?
I have some very questions also. So when you do the free energy, like you have a molecular dynamic simulation, like where the molecule can go anywhere on the protein, but
Yeah.
then when you do docking, you have a single pocket. And again, so I'm wondering, like, do the protein on which you did your work are known to have like a single pocket where like binding almost always happens, or like why?
Yeah.
Yeah.
So usually we just use so for the simulations, we just use the part of the protein that has the pocket. But since we kind of seed the ABFE with the starting pose generated by docking, so we dock the ligand into the protein, and then we use that generated pose to start ABFE. So usually, if the ligand drifts away from the site, it's not going to show good binding, even if it drifts away and somehow attaches somewhere else. It's only really going to show good binding if it stays in the pocket that we think it should be in, essentially.
Thanks a lot for listening to this presentation. We learned a lot today about MFBind and about how we can improve our models just by combining higher fidelity, more expensive data with lower fidelity data. And I think like the results that were shown are great and really puts us towards a path where machine learning can make more accurate and more reliable estimations.